Hi, I'm Annie,
Hi, I'm Steffi.
and today we're going to be talking about the analysis of the AT3G06130 gene in Arabidopsis thaliana. So the main two research questions we were focused on this semester were, what is the function of our gene of interest, and what are the genotypic and phenotypic differences or effects of a given mutation on our gene?
So the purpose of these labs were to understand the function of our gene in the Arabidopsis thaliana, as well as attempting to contribute to prior research regarding the organism. We also wanted to design and conduct our own experiment in order to determine what tissues our gene was expressed in, as well as what conditions affect the expression of our gene.
To identify our gene and its function, we first used an ab initio approach, which involved using the FGANESH software program to predict the exon boundaries of our gene of interest. Then we used the homology-based approach, which included using phytosome to find express sequence tags and the EST2 genome software to predict intron and exon boundaries. Then we generated a phylogenetic tree using the MAFFT program to help us hypothesize the function of our gene. To examine the genotypic and phenotypic effects of a given mutation on our gene, we used a variety of research techniques, including but not limited to DNA extraction and quantification, PCR reactions, gel electrophoresis, nanodrop, Sanger sequencing, and t-tests.
So basically, the next slides are about how we identified our gene and its function. So we used the ab initio annotation approach, and we were given the genomic sequence of the gene, which we had to put into the program FGANESH in order to predict the structure of the gene. Using this program, we mapped out the exons as well as the overall model of the gene. We found that exon 1 was 30 base pairs long, with the start codon ATG located at 525 base pairs, and the exon terminating at 554 base pairs. Exon 2 was located at 636 base pairs and extended 76 base pairs until 711 base pairs. Exon 3 started at 835 base pairs and extended until 2,150 base pairs and was 1316 base pairs long.
Using phytosome, we were able to discover the name of our unknown gene, AT3G06130. Phytosome also identified 4 ESTs, which are represented in dark blue in figure 2. These ESTs were found on chromosome 3 and were located on the minus strand. Using the ESTs generated by phytosome, we plugged the information into the EST2 genome software. Similar to FGANESH, EST2 genome also predicted that our gene has 3 exons, and it generated the exact same exon boundaries that FGANESH did.
We also determined which genes are closely related to our gene of interest and genes of other species than Arabidopsis thaliana, along with understanding how our gene may have evolved over time by creating a phylogenetic tree. We used MAFTT and narrowed down phylogeny from 100 sequences to roughly around 27 sequences. Each branch in the tree is related to our gene of interest, which is written in red on the map. Branches closest to our gene of interest signifies that these genes are closely related and could have similar functions, even if they are found in different organisms.
To further examine the possible function of our gene, we read some peer-reviewed articles on NCBI. From what we read, it seemed like our gene could be a part of the heavy metal-associated protein family. More specifically, some articles suggested that our gene could be a copper chaperone. Copper chaperones interact with heavy metal-associated ATPases to help maintain copper homeostasis in the cell. Thus, we hypothesized that our gene is some sort of copper chaperone that plays an important role in copper detoxification.
And so now we will explore the genotypic and phenotypic analysis of our mutants. So first we extracted the DNA from our Arabidopsis thaliana plants. And according to the data, DNA sample 1 was 192 nanograms per microliter, and the yield was 9.6 micrograms. The A260 over A280 value was 2.12, and the A260 over A230 value was 1.23. Since the A260 over A280 value is around 2, this means that there is a high RNA concentration in the sample. The A260 over A230 was 1.23, which means it is lower than the expected value. This could be due to the fact that there's a lot of carbohydrate carryover in the sample, and this is especially common in plants.
After testing the primers we designed with various DNA concentrations to determine the best PCR conditions, and after running the PCR products on agarose gel to see if the primers worked, we extracted DNA from our Salk mutants, performed PCR reactions on them, and ran them on agarose gel alongside our wild-type DNA samples. As shown in Figure 6, our control and 2 wild-type DNA samples all contained the wild-type allele, but none of the Salk mutants did. In Figure 7, we can see that all 10 of our Salk DNA samples contained the mutant allele, while the wild-type samples didn't. From this information, we were able to conclude that all of our Salk samples were homozygous mutants.
We then chose to send in our sample from well 8 of our gel electrophoresis for Sanger sequencing and chose to use our left primer as well, which produces a fragment size of 323 base pairs. We then analyzed the Sanger sequencing results that showed that the peaks were evenly spaced and the first position of quality sequence data was at 35 base pairs all the way through to 362 base pairs. The Sanger sequencing results also showed that there is little to no baseline noise present.
When we analyzed the Salk and wild-type potted plants, there were not many noticeable differences. The major difference was that there were a lot more Salk plants than wild-type plants, but this could have been because we accidentally poured more seeds into the Salk pot than intended. Another small difference was that the rosettes in the wild-type pots were tinted purple, while the rosettes in the Salk pots were more greenish-red. However, this variation was not consistent in all of the plants, so we could not conclude any of the phenotypes were due to the mutation.
We measured the average growth of the roots over the first 2 days, which was 0.8 centimeters for the wild type at 1 micromilliliter, and then 0.9 centimeters for the wild type that was not in the iron deficient condition, and 0.3 centimeters for the Salk mutant in the iron deficient condition, and then 0.7 centimeters for the Salk mutant in normal iron conditions. It seems that the iron caused an increase in root growth because both the Salk and wild type plants had greater root growth in iron rich conditions rather than iron deficient conditions. It also appears that the iron deficiency affected the Salk mutant more because there was a greater difference in the amount of root growth between these two treatments.
After exploring the EFP browser to see what kind of conditions affect our gene's expression and which tissues in the plant contain the greatest mRNA expression, we came up with an experimental layout to test the condition we chose. A paper published by Dinini et al. In 2008 used plant roots to examine the effect of iron-deficient growth conditions. Because there is not a lot of publicly available data that examines the effect of copper-deficient growth conditions, we thought this would be a worthwhile experiment to conduct, especially since we hypothesized that our gene could be a copper chaperone. For our experiment, we transferred pre-grown plants into a normal medium that contained all the necessary nutrients for growth and a copper-deficient medium. After letting the plants grow in these two mediums for 3 days, we collected the roots and ran qPCR reactions on them.
So our results from the RNA extraction quantification indicated that the lowest concentration was 28.1 nanograms per microliter for a copper deficient sample number 3, which is the image at the lowest right of the slide. We also found that the highest concentration of RNA was our copper deficient sample number 1 with 122.7 nanograms per microliter, which is the image on the lowest left of the slide. According to our A260/280 values, our RNA was mostly all pure with values around 2, except for the copper deficient sample number 3 and the copper sample number 2. They were both in the 1.8 range. This could be due to improper extraction methods and cross-contamination during the experiment. We statistically analyzed the results of the PCR reactions in order to determine if our hypothesis was correct. The results indicated that they were not statistically significant due to p-values above 0.05. Therefore, we cannot conclude how iron and copper affects the growth of the roots in the plant, as well as how it affects our gene. For the iron deficiency experiment, we got a p-value of 0.45, and for the copper deficiency experiment, we got a p-value of 0.41. However, looking at our bar graph, there seems to be a trend that occurs where copper treatment and iron treatment does seem to reduce root growth in our plants. These, however, were not statistically significant. In the future, these experiments could be done again with a larger population number to see whether iron and copper could regulate the expression of our gene of interest in roots, since there does seem to be a trend in the data.
In summary, we discovered that our unknown Arabidopsis thaliana gene is the AT3G06130 gene, and it contains 3 exons. The function of our gene is that it plays a role in transporting heavy metals, and this includes iron and/or copper. After applying a mutagen to our gene of interest, we discovered that the homozygous mutation negatively affects growth and increases chlorosis when grown in iron-deficient conditions. Our gene is highly expressed in reproductive tissues such as the carpel. Its expression is somewhat affected by iron deficiency and copper deficiency, but this relationship is not significant, so more experiments need to be conducted to confirm that this pattern is significant.
We would also like to thank our class and the teaching assistants, Brooke and Kira, as well as our professor, Sorrel Philly, as well. And thank you for watching.